Welcome back to the second episode in a three-part series having to do with CRISPR and the genetic marvels that go around that. But today, in this episode, we are going to discuss the complicated issues that go along with this technology. Now, if you haven't listened or watched the first episode, go back and do that. We covered how genetic engineering can help with certain diseases or pest control and infections that arise from there, or even agriculture. But let's not get ahead of ourselves because CRISPR is not just some magic wand. It is a very complex tool, and with that comes so many challenges. And today we're going to look at all the challenges that CRISPR has stirred up. So we're going to take a look at the ethical side, the technical side, and the social implications that are optimistically concerning. So yeah, let's get into it. CRISPR's precision is both its biggest strength and its Achilles heel. The method relies on guide RNA, or gRNA, to lead the Cas9 enzyme to a specific DNA sequence targeted for editing. However, the system isn't perfect. Off-target effect can occur when CRISPR edits unintended parts of the genome, potentially causing mutations in genes that regulate essential processes, like cell growth. This could inadvertently lead to disorders such as cancer. For example, in 2018, a Chinese researcher claimed to have used CRISPR to modify the genes of twin girls to make them resistant to HIV. While the stated goal was noble, critics raised alarms about the potential off-target edits and the long-term health implications for the children. And studies have since suggested that these edits could affect genes that likely are linked to cognitive development and even lifespan, a Pandora's box of unintended or unexpected outcomes. Another challenge is mosaicism, where not all cells in the organism carry the intended genetic edit. Well, when CRISPR is used in embryos, only some cells may end up edited, leading to uneven results. The issue complicates efforts to ensure consistent outcomes, especially in therapies intended for human patients. Let's talk ethics. One of the most controversial aspects of this CRISPR technology is germline editing, altering sperm or eggs or embryos, because the changes that are made are heritable, meaning that they are passed down onto the next generation. It may be amazing for something like Huntington's disease. It does spur a lot of really ethical questions for its use otherwise. In cultures far and wide, there are traits that are considered undesirable, and this could really lead to a slippery slope of eugenics. What if CRISPR could decide your height, your eye color, even your intelligence, or how your brain works? These things are what people are coining the term designer babies. And who do you think is going to be able to afford these designer babies? This is also known as genetic privilege. And what about the dilemma of consent? I mean, an embryo cannot consent to being genetically modified and having these unpredictable consequences as a result. I mean, even if the scientists and the parents have the best of intentions, there really is no telling what it will do to that baby and then generations down the line. Germline editing could inadvertently reduce our genetic diversity, which is crucial for adapting to environmental changes or fighting new diseases. And narrowing the genetic pool could lead to unforeseen events for our population. Think about like monoculture crops and like pests and diseases that just wipe them out. And while the United States have restricted the use of germline technology and editing to strictly clinical use, places like China and Russia are a little bit less stringent with its uses. While the World Health Organization is calling for transparency and requesting that these gene editing trials be recorded, it is a little bit sticky when it comes to what's going on here with enforcement. Let's zoom out and take a look at what's going to happen socially with CRISPR technology. Imagine a world where only the wealthy could afford to give these genetic enhancements to their children. This would create a genetic underclass. So that means that not only would there be, education and opportunity given to this wealthy class, but also biological opportunity. Now, historically, things like basic healthcare, vaccines, and life saving treatments are really utilized and offered to the wealthy because, well, they're wealthy. And these undeserved communities kind of trickle behind. Do you think that CRISPR could widen this gap or dramatically change how somebody gets social or biological or even economic benefits? And where one culture might see physical strength or beauty as being something to alter, another country or culture might think that intelligence is something that needs to be worked on. And it can only really further the divide between us and create this really kind of dystopian situation. Maybe. And then revisiting these environmental aspects that could be helped by CRISPR, like with the mice or with the mosquitoes that were mentioned in episode 1. What if disrupting those populations has more of a detrimental effect on the entire ecosystem, and we have no idea that that could happen? What if wiping out the mosquitoes on accident instead of just regulating them creates this trickle down or trickle up effect that really screws things up in the environment? And with all of that in mind, it should bring you a little relief that for now, CRISPR technology is really just focusing on helping with somatic cell editing. So that means editing within the body, and it won't be done in the germline. So it's not going to be something that can be passed down to next generations. It'll just stay with the person. Experimenting with this somatic cell editing is being used in diseases such as sickle cell anemia, where they are altering the blood stem cells in order for the body to produce normal hemoglobin. Hemoglobin. Anyway, another area that this is being used for is in the treatment of cancer. Like we talked about in the last episode, it is being used to actually ask the immune cells essentially to go ahead and attack the cancer. And I think that's pretty rad. But there is still the concern over accessibility because of the cost of these treatments. And while it is very noble about what they're trying to do when it comes to monoculture crops and pest control, the debate is still out on how it's going to affect the environment. It's something that we're trying to do to help feed the oncoming populations, but at the same time, that's an area that if we mess that up, whoo boy. I mean, yeah, go YouTube that. That's kind of a lot. And finally, one more complicated area that I just wanted to throw in there is this gene editing and the CRISPR technology being used to edit bacteria when it comes to either disease prevention or disease treatment. And that might help with antibiotic resistance. But then again, it might alter something that we don't quite understand. So yeah, it's kind of complicated there because we are already digging ourselves in a pretty deep hole and I can't get out when it comes to antibiotic resistance. I mean, that's real. And we have scientists everywhere. Even here in the Black Hills, we have a research team trying to figure out if they can do something about antibiotic resistance by looking at various types of bacteria underground over in the surf. But it's a very real thing. So it's complicated, but man, if we could get that to work and we knew that we weren't going to mess anything up, that would be pretty revolutionary. So, I mean, okay, so there you have it. Oh, the messy, the complicated, the fascinating world of CRISPR, along with its complications. And if you thought that all that jazz was complicated and like a lot to kind of deal with, well, just you wait. Because we are going to go into the dark side. We are going to go into the ugly parts of CRISPR that everybody's like, yo, what? It is a bit unnerving and a bit thought-provoking, if I do say so myself. So don't miss it. As we confront these controversies, these complications, these challenges, and we see the actual ugliness of what CRISPR can do. And until that third episode, stay tuned, be on your toes, be on the lookout, and I'll catch you on the flippity flop, my fellow nerds. Mwah.